Okay, let's discuss about it. So what is cancer all about? Cancer is a abnormal condition in the plants, abnormal condition in the organisms, in which what happens, there occurs uncontrolled growth of cell. In normal scenario, when you have a cell, this cell will be dividing into either 2 or 4, depending upon whether it is undergoing mitosis or meiosis. And as a result of that, after few generations, the cell will be dying off as and when the functionality gets completed, as and when it completes off its life cycle. You see the cell dying off with the process of a natural dying mechanism in cells, which is known as apoptosis. What is it? It is known as apoptosis, which is a topic which we are going to discuss after this cancer topic. Okay, so what is cancer all about? In cancer, or in cancer cells, you have a cell, it divides into two, let's say mitosis, then this will be dividing into two, and this proliferation, this division keeps on going. There is no end, there is no limit, there is no stoppage to this particular division. There is no programmed cell death. The simple name of apoptosis is programmed cell death, in which the cell, after completing its function, it will undergo or it will be dying off, right? So this is what apoptosis is, but which occurs in normal cells. All cells undergo, or all cells have the apoptosis ongoing, as a result of which you have the dead cells in the body. You have your dead cells on the in the body, that is, your hairs are dead cells, your nails are dead cells, your skin also like sheds off after a specific point of time. You see some kind of flaky skin on your hands, legs, skin, or on your face. So what is it? It is your dead cells that are getting removed off, right? In case of cancer cell, the programmed cell death is absent. You do not see any kind of programmed cell death, you do not see any kind of apoptosis ongoing, and as a result of that, the cells keeps on proliferating, right? So see, normal cells and cancer cell. In normal cells, you have, if the DNA damage is there, it will be dying off, it will undergo apoptosis, and the cell will go off from the main functionality. In case of cancer cells, the unfixable DNA damage, if it occurs by chance, let's say in the cell division, cell is not able to perform its functionality properly and it is still undergoing replication. So the cell will keep on continue to divide, and as a result, this problem, this uh, unfixed repair that's there, it still continues in further generations, and as a result, the functionality gets altered, right? So this is what cancer cell is, right? So what is it? It is uncontrolled growth of abnormal cells anywhere in the body. There is no set origin, there is no set area that only in this specific place the cancer can occur. The cancer can occur in any part of the body, in which abnormal like cells divide uncontrollably and they destroy the body tissue. These abnormal cells are termed as cancer cells, malignant cells, or the tumor cells. Over here, the tumors are formed. Lethal property is metastasis. What is metastasis? So you have a cell, or you have this particular mass of cell which is dividing uncontrollably, which is known as your tumor. Now some cells can break off from here and they can travel to some other location on the body and initiate cancer in some other part as well. So this property of the cell, or this property of the cancerous cell, to break off from normal area and to move to the other area and start off with the cancer, this lethal property is known as metastasis. Okay, so this is what metastasis is. What is metastasis? After your cell has performed the function, after your cancer cell has caused cancer in one of the area, it has the ability to detach from there, move to other location, and initiate cancer in some other part of the body, right? Spread of the tumor within the body. It helps in the production of secondary tumors in the new location in the body. It's a very, very lethal property. So that's the reason why even after the cancer patient have undergone chemotherapies, radiation, they are still under controls and checks in the by the doctors, where they can uh, where the doctor keeps on having a regular checkup to find out that there is no any secondary tumor there in the arising in the body, right? Other properties, in normal cells you see cells dividing and stopping after the the signal from the body, but over here many cell continue to divide and grow. As a result, you see the tumor being there. Then there occur variation in the shape and the size. Over here, nucleus is darker and larger than the normal cell. Over here, the number of chromosomes, the cell does not divide properly. The cell does not divide or replicate. The chromosomes also do not replicate properly, and there is no proper set arrangement as well. So abnormal number of chromosome arranged in a disorganized fashion, and over here the cluster of cells is not within a boundary. Like there's a haphazard boundary being there. Okay. Normal, and over here there is growth factor required in normal cell to divide, but for cancer cell there is no growth factor required. Presence or absence of the growth factor can cause will be still be causing the cell dividing, right? Then they are immortal, no apoptosis will be occurring. This is simple. So what is it? It's a genetic disease that is caused by the changes in the that are caused by the changes to the genes that control the way in which the cell divide, the phase function, and causes errors in the cell division, etc., etc. Right? Then basic properties. First one, uncontrolled growth occurs. Invasion and metastasis. Invasion and metastasis is they can invade the nearby tissue. They can cause cancer in new area as well. This is what metastasis is. Then you have the terminology of angiogenesis. So what is angiogenesis all about? Angiogenesis specifically means, because obviously, see over here, you have these cells being around, right? These cells are there. It's a mass of cell and it is growing undifferentiated. Like there is no set rule to it, uncontrollably it is dividing off, right? So these cells would any which way require nutrition, they would any which way require more blood vessels, more blood supply to cause the entire uh, growth and development ongoing, to keep the growth and development ongoing. So for that, angiogenesis, which is the process of formation of blood vessel. What is angiogenesis? Angiogenesis is formation of new blood vessels in the body, right? So around your blood, around your tumors, angiogenesis process is very very commonly seen, where the body or where the blood vessels are formed for so that the new growing cells or the new fast developing cells, fast developing tumor can have the nutrition. So this is what angiogenesis is. Then you have invasion of cell death. There is no apoptosis being there. The cellular mechanism obviously get altered, and immune system also is not able to recognize the cancer cells, like self cell and the non-self cell. These all are the various properties. Then you have metastasis done, hallmark. So hallmark means we'll be discussing in a bit about the various genetic proteins, genes, all of these things which help in the which are the main identifiers of cancer. Then you have cancer grading and staging. You all must have heard this particular terminology that every patient or every cancer patient is in a specific stage of cancer, stage 1, 2, 3, 4, depending upon the proportionality of growth, depending upon the extent or the intensity of the uncontrollable growth occurring in. You have various stages of cell. And what are the various cancer treatment? Then we can have surgery, there could be chemotherapy, there could be radiation therapy, there could be targeted therapies, depending upon different functionalities, depending upon different type of cancer, different type of scene can be there, different type of role will be therein. Okay, so remember about this thing as well. So you have uncontrolled growth, invasion, metastasis, angiogenesis, no apoptosis, cellular mechanism getting altered, immune system invasion occurring in. Then you have metastasis, hallmark of cells, etc., etc. Right? Did you understand till this particular pointer, everyone? Yes or no? We are discussing the basic properties of cancer. Right, let's move forward. So you have, and remember in the examination, you get a lot of questions from the various basic properties of cancer. And second topic from which you majorly get the question is from the genes concept. Okay, I'll be discussing about that as well. And to be really very honest, I would suggest you to not go into, see, cancer in itself is a subject to form, like it's a subject to complete, right? But if you look into the previous year papers and if you analyze the pattern of type of questions asked in cancer topic, it's very very specific. Okay, you get very simple type of questions. I'll be sharing those questions, like we'll be discussing those questions also in today's class itself. So try to limit down your present like preparation in the best of your ability, because that is something that will help you out in performing the examination better. Like it will help you to perform better in the examination first. Also it will help you to revise more and focus less on not important content. Okay, then you have common properties for resisting cell death, sustaining proliferative signaling, invading growth, activating metastasis, enabling replicative immortality, inducing angiogenesis, etc. Then, now one question which gets typically repeated, I'll show you just after this slide, and that is, on what basis do we define cancer type? Like how do we define a cancer type? So remember, the cancer is defined based upon the origin, based upon on which particular tissue is it occurring on. Or for example, breast cancer, for example, lung cancer, blood cancer. So you are defining the organ first, right? So it is based upon the origin, or it is based upon the, like, in which particular tissue is it occurring in, or on which particular cells are they occurring in. For example, carcinoma, that it means that the cancer which occurs or which develops on the epithelial cell. Then sarcomas, the cancer which develops on the bones and the soft tissues. Then you have myelomas, the cancer that develops on the plasma cells. Then you have leukemias, the cancer that develops in the blood cells. Lymphomas on the immune system. Mixed type, derivative from the multiple cells or multiple immune system tissue type. For example, blood cancer, lung cancer, etc., etc. Okay, so remember this thing, and then we'll be solving this. Giving you 1 minute for this, then I'll change the presentation and I'll ask you to go through the complete, or just quickly go through the question in itself. Read the concept, like read the question properly and mark the answer. Let me see how many of you can correlate the content shared to you right now. Give it a try, everyone. Very good. Everyone, try correlating the concept. We have just discussed it. Tumor type, the nomenclature. First one is a tumor that has arisen from the endodermal tissue. Then the cancer cells arise from the hematopoietic stem cells that do not grow as solid tissue. Then a permanent change. Permanent change means something that is occurring in at a permanent level. So that is a transformation, right? So C gets marked with sixth one. Then you have tumor that has arisen from the mesodermal connective tissues. Very, very simple. The answer for this particular question is very good. Option number B. So, tumor that has arisen from the endodermal cells, it is sarcoma, carcinoma, hematopoietic leukemia, then you have the permanent tissue change transformation, and then you have the tumor that has arisen from the mesodermal connective tissue, that is your sarcoma, right? Very very simple, very good. Out of 9 students, 5 of you participated and all 5 of you got the answer correct. Very good. Let's do one more. These are all the questions from the recent paper, like 2021, 2022. Okay, try this question as well, everyone. Let me see how many of you can get it correct. Try. Let's see. Simple again. You have again 4 options and the information related to the same. Tumor that has arisen from endodermal, then hematopoietic cell cancer that does not grow as solid tumor, tumor that develop from mesodermal connective tissue, then cancer that develop from the pigment producing cells of the skin. You need to find out the correct answer. Very very simple. So answer is option number 1 itself. Pigment myeloma. First one, D gets marked with the fourth one. Then you have leukemia, hematopoietic, sarcoma, it should be related to your mesodermal, carcinoma, endodermal, epithelial tissue, which we have just discussed, right? These are all 4 marker questions, and you can see very easily you can get the correct answer. Did you understand these 2 questions, everyone? Yes? Did you understand these 2 questions? Yes. Please make sure those of you are there in the class, all of you are participating, because you learn when you participate. Okay. There's no point just sitting in the class and not participating. If you're there in the class, utilize the time, participate so that you can get better role. You can get better understanding. Okay. Now you have the tumor cells. Okay. Tumor cells, you have primary growth also, you have secondary growth also. Right. So tumor cells, in the primary growth, we have cell to cell adhesion present, you have cell to ECM adhesion present, you have the cells getting localized. Cells are non invasive initially, then they'll be converting into secondary tumors or secondary growth, where the cells will become invasive. They do not release lytic factors, divide like normal cells, and apoptosis is absent. That's the first major criteria of your cancer cells. Then you have tumor cells, which converts or at a later stage gets converted into the secondary growth. So there is increase in the growth cycle, there is decrease in the doubling time, lytic factor released like metalloprotease, example collagenases, they get released. Then you have damage of the basement layer, it develop invasiveness, become isolated, contact inhibition is lost in these secondary growth, and then you have metastasis ongoing, that is the cell can travel to other part and cause cancer over there, right? You can easily get question from this segment as well. This is very easy. I'll give you a minute to go through this, then I'll explain you the gene concept, very simple again. And then like there are so many other things as well, but I'm just having a revision with you also. I'm touching the most important points only. Okay, quickly go through this, then we'll discuss about the gene topic, and then we'll discuss a question related to the same as well. Done, everyone, right? Now let's discuss about the genes that are involved in, right? There are two class of genes that are majorly involved in your cancer development. Okay, in the cancer, which are the two type of genes? So you have the tumor suppressor gene, and the second one are your oncogenes. Okay, understand these two and you will be you will never be making a mistake in the same. First one are the tumor suppressor gene, they're abbreviated as TSG as well. Tumor suppressor, something that suppresses the tumor. So tumor suppressor genes are the one which causes the suppression in a tumor. So these genes are initially inactive. These genes are initially active and they get converted into the inactive state in the or as and when the cell undergoes change and cause cancer. Okay, initially they are active because they are tumor suppressor. They keep the tumor in the suppressed state, right? And they get converted into the inactive state. And over here there is mutation also required in. What mutation and everything I'll explain. Wait for that. Just remember, tumor suppressor genes are initially active and they get converted into the inactive stage. Then you have the oncogenes. So you have oncogenes initially in the format of proto-oncogenes, and they convert into oncogenes. That is, from your active state or from your inactive stage, they convert into the active stage. Okay, so So tumor suppressor active to inactive, and oncogenes inactive to active, right? So remember about this particular thing. And in this, in first case, what is happening? Active to inactive. Is loss of function happening or the gain of function happening? Yes. In this active to inactive, which of them is happening? Is it a gain of function or is it a loss of function? Think about it, everyone. Just try to correlate with your own common sense. Very good. In this second, in the first scenario, in case of your tumor suppressor gene, there is loss of function happening in. And what about the oncogenes? Right? In the oncogenes, what is happening? Are you having the loss of function or the gain of function? Think again. Yes, in the second scenario, we have the gain of function happening in. Okay, remember, in tumor suppressor genes, they're initially active and they convert into the inactive stage. Then you have the oncogenes, they convert from the inactive to the active stage, right? Remember about this particular thing as well. Very, very important, right? So see over here. And one more thing, in your oncogenes, only one mutation is sufficient enough to cause the cancer. In the case of your tumor suppressor gene, two mutations are required to cause the cancer. Why? Because oncogenes are dominant and tumor suppressor genes are recessive. Okay, so remember the oncogenes, there is gain of function. There, these are dominant. They are initially inactive. They convert into the active format from proto-oncogenes to oncogenes. Any of the mutation can help out. Point mutation, gene amplification, gene deletion, translocation, transposon, any of them. Examples: RTK, RASMAP kinase, JAK-STAC, serine, synthionine, any of them. Then your other tumor suppressor genes, so require two mutations. Recessive mutation in both the alleles is required. There is loss of function, and examples could be anything. It could be P53, retinoblastoma, P10, etc. Right? Remember this as well. After this, try finding the answer for this particular question. If you have heard what I was explaining you in last 10 minutes, you will not make a mistake in this. It's a 4 marker that has been asked in the examination. Try the question, everyone. Giving you 2 minutes, read the question carefully and mark the answer. Let's see, very simple question. So you have two classes of genes, the proto-oncogenes and the tumor suppressor genes that contribute to the development of cancer. Following statements are given to you. First one is saying the proto-oncogenes result in the development of cancer by the gain of function mutation, whereas the tumor suppressor gene leads to the development of by the loss of function mutation. Then you have the proto-oncogenes result in the development by loss of function. What did we just understand? In case of your oncogenes, there is from inactive to active, so gain of function is happening in. So which one should be the correct one? Yes, proto-oncogenes, they are inactive. Then they convert or they have loss of function or gain of function. They have gain of function, right? From your inactive to the active state. And in the tumor suppressor gene, there is loss of function. So A is correct. If A is correct, D cannot be the correct answer. Then B is wrong because they're contradictory. Now the answer is A and C or A and D. Third option, mutation in both the alleles of the proto-oncogenes is required for the induction of cancer, whereas mutation in one is required for the tumor suppressor one. We have just understood in proto-oncogenes one, and in the tumor suppressor two of them are required. So A and C. B is the correct answer. You can remember it this way as well. Oncogenes and one mutation, both of these starts from O itself, and the second one is tumor suppressor gene and two mutations, so T and T, O and O. B is the correct answer. Mutation in one of the allele is required for the proto-oncogene is sufficient, so this one is correct, and in both of them is required for this second one. So what should be the correct answer? Answer should be D is the correct answer. So C is the correct option. Sorry, C is wrong. This is correct. I just read it. I have explained the correct thing, but I just marked it wrong. So A and D are the correct statement, right? C is the correct answer. A and D statements are correct. Did you understand the question, everyone? C is the correct answer, not B. Is the question clear to all? Right, this is about this particular question. After this you have the various genes, the examples, where do you find them, that as well. Molecular biology I've already seen. If you want to go in the depth, you have this entire table where the selected oncogenes, their mode of action, associated human tumors, everything is written down. See that. Right, after that, this is normal. Yeah, there were many other questions that were asked. We'll be doing them in class itself. Okay, so I've taken up very like whatever is important from cancer topic, right? Please make sure that you're going in depth as well. If you do not have studied, like, because it's a revision class, so I cannot go in like major depth. So I've taken up, or I've like highlighted all the pointers, right? This was an extra short class for you all.